seminars. We are honored to have you, ma'am. Thank you. Over to you.
Thank you, Avantika, and welcome my young colleagues and teachers. It's again we meet today, and I hope that all of you have enjoyed the seminar. And today is the last talk. So, but I see only very few participants, Grace. Only 15 of them. Or are they all seeing the live streaming? Maybe that may be the reason.
Probably, ma'am. And we just broke for lunch, so.
Okay. No issues.
They will join, ma'am. They will join.
Sure. No problem. So let me share my screen, and then I'm going to tell you a very different story today, and also give you a glimpse of how a simple talk over a cup of coffee or during a meeting can in fact lead to some very big discovery. It is just the same like if you all are about how the Nobel work of CRISPR-Cas won the two women, that is Emmanuel Charpentier and Jennifer Doudna, to get the Nobel. So in fact they were in their two different fields, both definitely working on a different aspect of CRISPR and gene editing. But it was just in one of the conferences, the ASM conference, American Society for Microbiology conference, where they met over a cup of coffee in a cafe in Puerto Rico, and everything developed. And it is in a history, it is the shortest time that was required for both of them to come together and come up with this gene editing technique, which won the Nobel. So many things can happen. So I was giving a talk about this Nobel lecture in University of Davangere, and I titled it as, anything can happen over a cup of coffee. So it is so true that these dialogues sometimes can click, and where it will click, we don't know. So I am going to, so some of the other professors, they were joking that this can be one of the best caption for the coffee day, cafe day. And they have something like that, for over a cup of coffee. This is a similar story that I'm going to share with you. So my young colleagues will really get inspired and would definitely try to grab the opportunity. So now let me share my slide. Grace, you have to give me permission to share my slide. Oh, already given. Thank you so much. Yeah, thank you. Can you see my slides, colleagues?
Yes, ma'am. We can see your slides.
Yeah, sorry, I was waiting for your answer. Okay, I've put it in the full screen mode. So this is a story.
It's there, ma'am. It's there, ma'am.
It's there, na? Thank you. Thank
Yes,
you so much. Yeah, thank you. Kindly feel free to stop me anytime that you would like to and ask questions, or in the end, put questions in the chat box, etc. So the title, if you see, it is Shocking the Biological World. Now, I'm a microbiologist. My expertise lie in understanding the infectious disease by bacteria, and of course by viruses and by parasites, but mostly bacteria. And this kind of very unusual collaboration between a medical microbiologist and an aerospace scientist could not be imagined until and unless we indeed came together in one of these meetings, and this particular collaboration took place, due to which the amazing medical applications have come into picture. Now, how this unusual collaboration began? This is Professor Jagdish Gopalan, who is a professor of aerospace engineering, and myself. We both were student council advisors. And we had a meeting, we were having a meeting. So, as usual, that when you gather for a meeting, some of us will come little earlier, and then we will wait for the others to come. So I was new, I joined that time. It was, I think, 2005. So I was waiting, Jagdish joined, then he just greeted, exchanged greetings, etc. And just asked, what are you working on? And I was telling, so this is my area, etc. And I asked, what are you working on? So he said, I am an aerospace scientist. We work on the shock waves. And so he explained me the propagation of the shock wave, etc. And he also told that, see, Dipshikha, it is a very interesting phenomena of the shock wave, that you can it can create a devastating problem like bomb blast, or if you can really tweak it, it may give rise to something very, very fruitful. But I do not know how to put it. That's what Jagdish told me, and from then on there was no looking back. Is the first student, Dr. Sandeep, who is, I was mentioning to you about him, and he's a medical doctor, and he was the first person to actually visualize this single bacterium in a vacuole. And he is actually also having a very different mind. He started asking that what happens to the virulence of a bacteria when you take it to the space, to the outer space. In the meantime, when we were thinking, when we were discussing, because the outer space microbial or the life form, it is still a big mystery. There had been a lot of experiments that have been done. They have been told by the by ISRO, etc. There were too many programs on the outer space biology. But the question was that if you send a bacteria to the space, what happens to its virulence? So that's how also we started talking to Jagdish, and among all the aerospace scientists, he was the first one to respond to us. And that is how he initiated this collaboration in 2008 with another aerospace engineer student, Dr. Rakesh. He is currently the dean of Amrita University. Now, the first PhD student, the joint PhD student, Dr. Divya Prakash, and second PhD student, Dr. Akshay Date, they both actively got involved in understanding the biomedical application of shockwave. So it was indeed a very small discussion over a cup of coffee in the meeting room, which led to now a more than 10-year-old, beautiful story. And the story is so successful that we are very soon going to wait for the launching of our diabetic foot infection healing device. The trials are awaited, and we got this imprint grant of DST, which is really one of the major grants to promote the medical devices. Now, what I'm going to do is I'm going to give you a brief introduction of what are shock waves. Some of you had your physics in 10th and 12th, and you may actually remember some of the concept of waves. So, in fact, what are shock waves? Any disturbance in a medium which produces the compression wave, which propagate at the local speed of sound. And the beauty of the shock wave is, and how do you define? Actually, the shock waves are the non-linear waves that are formed by coalescing of many such compression waves and traveling at a supersonic speed. That is the speed which is greater than the local speed of sound. And shock wave, which are produced by the sudden release of chemical, mechanical, nuclear, or electrical energy. So under many conditions, I will show you a couple of examples. One can generate a shock wave. See, in 1922, way back in 1922, Becker have given the profile of the shock wave. So suppose this is the piston. It is like a piston. You just imagine the piston or the pichkari, with which you play holi, etc. Or you just push in water. Just example, take that example, keep that example in your mind and see that this is a piston, and this is the one which is creating a pressure and pressing this air out. Now, this kind of pressure comes in two different forms. The first is the blast wave, and second is the shock wave. Now, the beauty of the shock wave is that the shock wave profile, if you really look, you have a phase where the pressure is increasing and then it becomes a stationary. Now, this particular phase is extremely important for the biological applications. Unlike blast wave, see what is happening in the blast wave. You have the very rise of that is amplitude of the pressure wave, and then the standing time here is extremely low. That means there is a very high pressure, and then there is just a rapid fall. And the fall time is extremely rapid, and you do not have this so-called standing duration. What you have it, the C time. So if you look into the duration of the pressure pulse, the C, this particular part, this is the one where majority of your biological signaling and processes is happening, which I am going to discuss with you today. Blast wave is highly degradative in nature. That means it will demolish anything and everything. You know about the atom bombs. You know about the blast wave, or which is used to blast off rocks in the many sites. So this is a very detrimental wave. Now, the same detrimental wave can be somehow manipulated in a way that it becomes a much useful shock wave with a better duration of the pressure pulse. So this is to be taken into account. It cannot drop like this very rapidly after giving such a high amplitude. And how do we play around with these particular pressure pulse duration? I will also try to tell you a little bit about the basic of the shock waves, because some of you may be, very much interested to work much later, not now, in the interface of chemistry, physics, and biology. Many of you may be interested to do your PhD in the interdisciplinary area, which is coming up in a very big way. And as I always tell all my young colleagues, that biology cannot be done without the understanding of physics, chemistry, and mathematics. So my dear colleagues, kindly do not leave these three subjects any time out of your scope, because it is extremely important for you to ask a question which is really out of the box. So anyway, the basic of the shock waves is by these three principles. That is the surface of discontinuity, compression pulse, and the intense sound wave. So once this Vasiliev and Stanukovich mentioned that one could say that shock wave were discovered for signs like planet Neptune and Pluto at Penn Point. It's a very kind of a philosophical term. And August Toepler was the first to experimentally visualize the shock wave. What are the differences between the sound wave and the shock wave? And the differences are marked. Say, for example, sound wave, the process is reversible. But the shock wave, it is an irreversible process. In sound wave, there is a change in the physical state of gas. The changes in the physical state of the gas, they are extremely minute. But in shock waves, there are very large changes which are induced in the physical state of the gas. And also in the sound wave, there are very small amplitude longitudinal displacement of the molecule, whereas in shock wave, they are carried in the direction of the propagation. So for example, now see this supersonic bullet. So suppose if you fire a pistol, and this bullet goes into the supersonic speed. Now with a very high speed camera, the scientists, including our lab, with Jagdish's lab, they're able to measure the so-called wave front. You are seeing this? So this is these ripples. This is the shock wave which is produced, this is called as a wave front. So the molecules are carried in the direction of the propagation. Whereas you see this is also secreting this so-called blast wave. It's a big blast, and you see this halo of the wave, which is being progressing. So the shock waves becomes side of the two sides of a coin. On one hand, it can be extremely detrimental, but if you can think very properly, you can in fact turn this shock wave into much useful wave, which can be used for your biomedical applications. Now, indeed, is shock wave a boon or a bane? How can you make it the bane from to a boon? So there are many questions that one can ask. Now, where all these shock waves gets generated, so you will have a wonderful examples in the nature where you will see the examples of the shock waves getting generated. Say, for example, if you look into the natural sources, you have the thunderclap, the volcanic eruptions, the earthquakes, extraterrestrial, the cosmic explosions, the meteorite impact, the solar wind, this is from this Hubble Space Telescope. And you look at here, this is lightning. All of them generate the so-called shock wave. But you have a man-made source of the shock wave. Before coming to this, let me also show you. Look at this. This is a courtesy United States Atomic Energy Commission, where you see that the shock wave. Can you very nicely look into these ripples of the wave that is being produced by the wind, which is in fact, taking away all these trees. So this is the shock wave. And this is the Atomic Energy Commission, where these ripples, what is created is actually creating this shock wave. So the explosion of an atom bomb this particular system generates a massive shock wave which carries about 50% of the total energy of explosion. Wind blast, this is the wind blast. This traveling at a speed with an over pressure of 5 Pascal can destroy a 2-storied brick house. Now this is the same wind blast which is coming, right? And now it can destroy a 2-storied brick house. So that much is the power of these waves. Among a man-made sources, I will tell you a very interesting example of a whip cracking. Now, whenever as a child, all of you must have gone to a circus, and you might have also noticed that the ringmaster always comes with a kind of a whip. And the moment he cracks the whip, the animals obeys. Now, in fact, the whip never ever touches the animal, be it tiger or be it elephant or be it a bear. It never touches the animal. It never hurts. But what happens is that cracking or lashing of the whip creates that shock wave, and the shock wave travels inside the body, which freezes the animal. And that is how the animal once learned that this is the task that the animal needs to do. Say, for example, the ringmaster say you get out from the the ring which is on fire, or you stand up on the stool, etc. That is the indication, because the animal freeze, so they learn very quickly. So this is the scientific mechanism behind the whip cracking, and how these animals, which are supposed to be really very kind of dangerous if they are open in the wild, become so tamed or so domesticated. Now, clapping of the hands also generate these shock waves, which you can record with a high-speed camera. The musical instruments, of course, the shock tubes, which are the expertise of the lab of hypersonic shock wave of Jagdish, and where he has put many shock tubes in many colleges, and now the shock tubes have become a very important part of the syllabus for aeronautical engineers. Supersonic ballistic objects, supersonic aircraft, and explosives. So these are all man-made. Now, having all these examples in mind, the question comes that how the shock wave now can be tuned, and can we have it somewhere applied for the mankind? And indeed, it is true. Now, sky is the limit, because anything that you can think of, by perturbing the pressure profile or the pressure pulse, you can make it a use for something else. It started from making a shock tunnel in the aerospace engineering lab to the blast mitigation studies. Sorry? Okay, no question. The chemical kinetic studies, the chemists have used the shock tunnel to understand the chemical reactions under certain conditions. Then in our lab, we have profusely worked on the transformation of the cell, transformation of bacteria by using a shock tube. You know that the electroporation is a technique to introduce the foreign DNA into the bacteria. And the electroporator, the machine electroporator by Biorad, it's very expensive. Some 5-6 years back, the machine itself used to cost around 3 to 4 lakhs, and now the cost has increased to 7 lakhs. Each cuvette will cost you somewhere around 500 to 600 rupees, which is use and throw. So the entire cost of one transformation goes dramatically high. So what we did was we used this the shock tube, and made this shock tube to transform the bacteria. So cell transformation was very beautifully done. And then there are the Murugappan tea company, they use the shock wave to improve the flavor of the tea. Because when they use the shock wave, there are certain polyphenols which got exposed. And that is where the aroma of the tea, that completely became very much available. So it was very different. So when our Prime Minister visited our institute, I think some 6 to 7 years back, and we have given him this tea, and the Murugappan company chair was also there, and they borrowed that technology, and now they are utilizing it for improving the tea quality. Now we are working extremely closely with the medical team to understand this wound healing, and this is what I was talking about, especially in the diabetic foot infection, where the healing of the wound is extremely difficult, and it is a very sad state of affair where the amputation is the only way by which one can save the life of the person who is having already a diabetes, then the diabetic foot leading to the gangrene. So it has many branches, and these branches will never stop. As and when we discover, right from mathematical modeling to wood science to food science to petroleum oil extraction, almost everywhere you can have this technology applied once you work on the pressure profile. How did it all began? In fact, again, it is always a beautiful story of the vital few. Vital few means you don't need a big mass to do something, but you should have a person who are very like-minded, and they are always few in number. So this is called as a theory of the vital few. And always when it is a theory of the vital few, the things becomes very, very promising, and something comes out remarkable. Here it's a story of Eberhard Hausler, a physicist, and a Francisco Lorando, a urologist. So a doctor and a physicist. They came together and started the use of the extracorporeal shockwave therapy. This extracorporeal shockwave is nothing but an underwater shockwave, and to treat the brain tumor. So this is the first patented design of the extracorporeal shockwave in 1947, where they can target these shockwave just to the tumor area and try to treat it and regress the tumor. It's extremely successful, but till now the use of the shockwave, which already got the FDA approval and which is used very commonly, is a lithotripsy. Now, the kidney stone is a very common kind of a disease manifestation in many individuals across the world, and this can be life-threatening. If the stone is small, then it can be forced to expel by many mechanisms. But suppose the stone becomes bigger and bigger, then it blocks these tubules and venules of the kidney, which can lead to the kidney failure. And the only way it is to do is to remove them. So the surgery is one option, but now this lithotripsy is the one which is majorly used, where you put, in fact, the person, I will just quickly show you how the lithotripsy is done. This is the person is put under water, and this particular kidney is targeted with these shock waves. This is called as an underwater shock wave. Why you use the extracorporeal underwater shock wave? Because the waves, the pressure, it gets attenuated, and when it reaches the kidney, it should be disintegrating the stone, but not damaging the other organs. And this is an extremely expensive instrument. Dornier lithotripsy, there is one company in UK which makes this, and they supply to all over the world, and they are the one who are actually a pioneer in making this lithotripsy. So, and this use of these extracorporeal shockwave lithotripsy, in fact, has certain other issues. And the issues are, suppose now the stones are broken, but where are you dispersing the stones, the fragments? Can the fragments go and then again form like a nuclei and make more stones? They made one very beautiful attachment or add on to this system, where not only that the shock is given, but there is a very fine catheter which goes, and like a suction pump, it sucks off all the disintegrated particles, and it takes it out, so that you don't leave those particles elsewhere to go and nucleate and form another stone. But the problem with this extracorporeal shockwave is, if you really look, the profile is just like that of the blast wave. It's extremely high pressure and focused wave, and it is the fall time. It's a very rapid fall time, and then again you see this small pulse. Now, this kind of very high pressure pulse and the focused shock wave can have a collateral tissue damage. Now, that is the reason that why these extracorporeal shockwave, underwater shockwave, was not very much favored by the medical community. And you do have a lot of complications. So, for example, the tissue effects, renal, cardiovascular, gastrointestinal, genital system, especially in the fetus, and one may have a renal function dysfunction, delayed renal function, hypertension, the fertility issue. And all is because the pressure itself is so intense that it can do do too many things to the body organ. And that is the reason that such high impulse shock wave cannot be used for various biomedical applications. So how do you tweak it? And here comes our two lab. This is the laboratory for the hypersonic shockwave research. This is the one of the largest lab in the Asia. I would say second largest in the world after Japan, because the originators of the shockwave tunnel, Professor Jagdish and Professor Reddy, they are IISc. So they are the one who are the pioneer in setting up the shock tubes across India, across the world. So this is our hypersonic shockwave lab, and this is our molecular pathogenesis lab. And we came together and now made a wonderful devices and other biomedical applications. So you are welcome to actually come and see this lab. It's a huge lab. You see these different shock tubes. So these are all different shock tubes which are made, which are by the team, and they are assembled here. So a very, very high-end experiments goes on, from supersonic scramjet aeronautical questions to the biomedical applications. Now, the conventional shock tube. So suppose if I am telling you that this, what is the difference between a conventional shock tube and the diaphragm-less shock tube? Now, the concept is very simple. You don't have to, pay much of your energy in understanding. You see, this is a long tube, and there are so many outlets here, right? Pressure valves gauge, pressure monitoring gauge, temperature monitoring gauge, lot of sensors are mounted, and that is how it is looking. You can make your own shock tube. You can vary the length of your shock tube, and that is what we do in the experiment. But what happens in the conventional shock tube is that the repeatability is affected by the varying nature of diaphragm rupture. Say, for example, now this is the piston. Yeah, so you are pushing the piston. And the piston, when it is getting pushed, the shock wave is traveling in this driven section. There has to be an end here where your material needs to be placed. So the material can be anything. The material can be a bacteria with a DNA. Material can be a vaccine which is ready to be injected into the animal, or the material can be a chemical where the chemists are trying to understand the chemical reaction. Material can be anything at the other end. But what happens is, when the shock wave is traveling with such a high pressure, to attenuate that, there is a diaphragm. So the diaphragm gets ruptured and then the shock gets released. So this is the with diaphragm or the conventional shock tube. Now, the diaphragm shock tube has an issue because the diaphragm material itself, you can have a paper diaphragm, or you can have a rubber diaphragm, you can have a metal diaphragm, depending on the use. The diaphragm material itself will contaminate the shock tube, and it may damage the internal surface. And replacement of the diaphragm will be very costly and very time consuming. All the time you have to replace. So one shock you give, do the function, replace the diaphragm. So that can be very cumbersome. So for that, a new shock tube that came to our mind was a diaphragmless shock tube generator. That means there will be absolutely no diaphragm. It will be directly, the shock will be generated and the shock will be completely delivered to the medium without the aid of the diaphragm. But one very important point that you need to keep in mind is that one must have a very good idea about the pressure profile. And that is why so many sensors, if you really look all across, there are tiny sensors which are mounted, which measures the pressure at each point, so that the pressure is the critical point here. Too much pressure is the issue, and too less pressure will not do the function. See, our first very good diaphragmless shock tube, which we made modifications due to our biological input from the biologist, is that this kind of an L bend. Now, when we place the mouse in the shock tube, the mouse became quite, kind of scary, kind of stressed, etc. Because before we had a closed chamber. Then we modified the chamber, we made it aerated, we allowed it to, have the light from all around, and the mouse was very comfortable. And this L bend, in fact, is important because the shock is traveling. This is the driver section. So you push the trigger. Shock is traveling, and then in the L bend, it gets kind of attenuated. So whatever is getting delivered to the mouse, that is far, far less pressure profile than what you start with. And that is the one which is important for the biomedical application. So these are all the pressure signals that you see, which is measured across, and what is also now we are trying to measure the pressure signals that is actually felt by the animal. So with this, actually the in vivo experiments, they became very, very fruitful, and we could do a lot of our in vivo experiment. The first example of the work in the lab, which led to the generation of a non-tube shockwave generator, which led to the discovery of the needle-free vaccine delivery system. So this is the hallmark discovery of our lab, where you have tubes, and this is how the machine looked. So this is the trigger. You press the trigger, the pressure it travels all the way through this nonal tube, and the end what you're seeing is this cavity, and this cavity contains the bacteria, the DNA, or the vaccine to be given to the animal. Now this particular device also worked like a electroporator, and the transformation efficiency was just amazing. It was more than 70 to 80% of the transformation by this by this shockwave generator, our own homegrown shockwave mediated electroporation, rather than the biorad electroporator. So we have a lot of comparison between the two. And the system is very simple. You have a nonal tube and you pass. But the question is, which is very important, and we had to discontinue, that this nonal tube was coated with an explosive, this HMX, octahydro 1357 tetranitro 1357 tetraozyme. So this is an explosive, and the spark, it makes this explosive generate this so-called the pressure, and then this is the there is a diaphragm, and then the diaphragm burst, and the material whatever the material is there at the receiving end, they do their function. Like, say for example, DNA gets into the bacteria, or vaccine gets into the skin. But again, the problem is that FDA would never give us approval if you have this. So we had to get rid of the entire nonal tube. And the major challenge was that how do you replace this nonal tube? And what do you do again to make your next generation? This is the solar powered miniature shock tube. We got rid of everything. We got rid of the nonal tube. We got rid of any explosive material. This is a very, very beautiful solar power operated, the next generation shock tube generator, which we use for the diabetic wound infection healing. And the principle here is very simple. The principle here uses the two gases, hydrogen and oxygen. When they are mixed, they generate this so-called pressure. And these particular hydrogen oxygen generated pressure, the wave travels, and the end product actually is a water. So it is an highly, highly desirable system. Of course, with a lot of care, you need to make this system ready. So this has been manufactured. Now we are trying to miniaturize it much further. So the high pressure and the temperature, that needs to be very beautifully calibrated for its use in different biological application. Say, for example, transformation needs some kind of a pressure profile. Diabetic wound healing will need different kind of a pressure profile, whereas the biofilm disruption will need different kind of a pressure profile. So all these are, very heavy on the experiments to, in fact, standardize the equipment. And once we standardize it, we keep that pressure profile constant, and there is no way that anybody can tamper with the pressure profile. There is one more addition to this shock tube, that is a conical shock tube. Now, this is the latest shock tube that we have in our system. In 2019, this shock tube was built. And the beauty of this conical shock tube is that you have from a very low pressure to begin with and concentrated into this high pressure. Now, every material is kept at the end of this conical high pressure tube, and then you have the material, the mouse or the vaccine strain, etc., whatever that you would like to do at the end of the tunnel. This conical shock tube at this moment is a bit bulky. So if you see this, so this is an example of the conical shock tube. This is the mouse, And we have kept the mouse here with these, kind of a strainer, so that the mouse is comfortable and it's not moving around. And then we did our experiment to show that how the diabetic wound healing or the vaccine delivery happen along with that. So this is the latest generation, and a lot of experiments are now getting done with this conical shock tube. Now, let me tell you, before coming to some examples of the shock tube and its role in disrupting biofilm, all of you are a student of microbiology, majority of you, and you would love to, and I am sure you know a lot about the biofilm. Now, biofilm is a term which we use very loosely. It is nothing but a film, which is formed by a bacteria. And but the biofilm is an extremely complex, dynamically complex biosystem, and the bacteria lay down this biofilm. So if you look here, so these will be a planktonic bacteria, then they will come together. There is a crosstalk which is going on between the bacteria, and once they reach a correct quorum number, or the quorum sensing happens, then they form this biofilm. There are more than 25 to 30 genes which gets upregulated, and this decision allows them to make this biofilm under certain conditions. Specifically in the surgical implants, in the pacemakers, lung tissue, gallstones, they all form these biofilms in the base of the ship, and the ship can get corroded and the entire ship can sink. So that dangerous is the community behavior which is exerted by the bacteria, which is none other than this biofilm. No amount of antibiotic is going to be accessed by the bacteria if you have this biofilm which is laid around. So any amount of antibiotic will be futile, which will lead to another problem of generating antimicrobial resistant in a population. So we will keep on dumping the antibiotic, but nothing will work. And one very important point about the biofilm, which we are trying to understand, and now medical community is also getting very much aware of this, that every bacterial disease actually starts with something known as a collective behavior or microcolony. And this microcolony is nothing but an initiation of these so-called biofilms. So these are biofilm infections. So you can take an example of somebody having a bad sinusitis, where there is actually no cure for sinusitis as such. And some may have also realized or noticed that you can go on taking antibiotic, but the sinusitis pain, it never goes away. It is a very different kind of the pain. Some of them are bacterial mediated. The sinuses get blocked. And this is because the bacteria, it completely forms a coating, that is a biofilm. Until and unless you break that, there is no way by which any antibiotic can act on. So lot of these infections, the lung infections, the gallstone infections, etc., are because of the laying down of the biofilm by bacteria. What can be the sites of these biofilm infections? So all these medical implants, for example, central venous catheters in the central line when they draw, the prostheses, the pacemakers, a cardiac implant, artificial joints, hip implants, artificial knee joints, or the urinary catheters, each one of them, if there is something implant has been put, they can be a site for the generation of the biofilm. So biofilm mode of infection can be very life-threatening, especially when it comes to the organs. The model system that we have used here is Staphylococcus aureus. This is a gram-positive bacteria. Again, an important nosocomial cause of infection. And Pseudomonas aeruginosa, which is a gram-negative bacteria, is an opportunistic pathogen and is now one of the most important pathogen in the diabetic foot infection. So both of them, Staphylococcus aureus and Pseudomonas aeruginosa, along with one more bacteria, Acinetobacter baumannii. These three indeed create an havoc in the diabetic wound and make the wound kind of a chronic wound. This is a non-healing diabetic wound. And as I mentioned to you, the only way by which one can take care is by amputation. So this is a serious matter. So we use these model organisms and try to understand that how can you use the shock wave to disrupt the life of these organisms under infection conditions, and also prevent the bacteria to have drug resistant phenotype. So this was important. Now, if you look into the biofilm which is laid by all the three bacteria, Salmonella, Pseudomonas, Staphylococcus aureus. All these trees have a very important way by which they lay down their biofilm or the community behavior. They have a collective behavior. This is a scanning electron microscopy of these 3 bacteria, which are beautifully forming a biofilm. The moment you use the shock wave and give this bacteria the shock wave, you see that the entire biofilm is disrupted, showing that indeed true, the biofilm can be disrupted. The sturdy biofilm, on which nothing acts, no antibiotic, no detergent, nothing will act, shock wave can lead to the disruption of the biofilm. Very good. And if you really look into the OD of these bacteria with the shock wave, you see that a control versus the shock wave treated, it drastically had fallen. So indeed, the shock wave was able to disrupt the biofilm. Now, in vitro is fine. What happens in the in vivo situation? Now, Pseudomonas aeruginosa, though it was known as kind of an environmental pathogen quite some time back, now we know that majority of the death that is caused by that is caused in cystic fibrosis individuals is actually not because of the cystic fibrosis disease, which is a genetic disorder, and they have a mutation in their transconductance channel, CFTR channel, and they cannot clear the mucus. So there is a huge amount of mucus that gets accumulated in the lung of the cystic fibrosis individual. The death is not because of the huge amount of mucus. The death is because the Pseudomonas aeruginosa takes advantage of this mucin-rich environment and they grow profusely there. And the death is due to the overgrowth of the Pseudomonas aeruginosa in the lung. So we mimicked the model and we used the intranasal infection model, and the bacterial colonization in the lungs were checked. And then we asked a question whether now the shock wave can break the biofilm. So once the biofilm is broken, let us give a small amount of antibiotic, then which will act on the planktonic bacteria to clear it off. Let us see how the experiments are designed. You have the Pseudomonas infection. So we use an intranasal infection model. And after 4 days, it is either left untreated, the mouse are left untreated, or they are treated with ciprofloxacin alone, combination of shockwave and ciprofloxacin, or shockwave alone. And then we have to euthanize the mice and then look at the lung CFU and also take the electron, the scanning SEM picture of the lung, integrity of the tissue to understand the integrity of the lung. So look at the CFU per gram of the lung. The control, they have an extremely high number of the bacteria. Shockwave alone does not kill the bacteria. It breaks the biofilm, but it does not kill the bacteria. Now, ciprofloxacin alone, this is an antibiotic, remember, against which the Pseudomonas are very sensitive. But in a biofilm mode, no amount of ciprofloxacin is going to kill. It's absolutely no difference. But the moment you give shockwave plus ciprofloxacin, the CFU comes down, showing that making their bacteria come out of their biofilm mode makes them really very insecure in a planktonic niche, and then they are attacked by the antibiotic, that to low concentration of antibiotic, and the number comes down. How is it reflected in the terms of the mortality? Let us understand. Now, this is the SEM picture that I was mentioning to you. No infection, the lung look like this, very, very clean. Then you have the bacteria which is colonizing the lung. Look at this beautiful, the biofilm which is formed by the bacteria. Shockwave alone, there is a disruption, but there is absolutely no killing. Ciprofloxacin alone, absolutely nothing is budged, nothing is changed, and because nothing can enter, so what will change? So they also show just typically like that of the bacterial infected. But the moment you give them a shock wave and then a ciprofloxacin treatment, you see that these two lungs have become now very much similar. That means the shock wave made the biofilm disrupt, and the bacteria came out of their niche of their house, which were then attacked by a ciprofloxacin. So this was a very wonderful attack strategy to save the host. And what is the resultant percentage survival? It's very encouraging and amazing that when you have shockwave and ciprofloxacin, you see 100% survivability, which is not there in either shockwave alone or ciprofloxacin alone, showing that the biofilm mode of infection is one of the important mode of infection that we need to think, and simply dumping antibodies may not do anything good. The same philosophy was used because after we published this literature, there were a lot of dentists and the MD medical student, the dental students, they become interested because for them, the dental biofilm is a serious matter. You know, it's a huge burden on the dental society because of the biofilm that is formed on the teeth, and which then actually destroys the teeth to no tomorrow. So we got an MD student who came to our lab and did his MD thesis with us. And we figured out, and these are all the tooth samples that he extracted from a KLE. He was doing his MD in the KLE Dental College. And you see here, this is the control dentine tooth with all the bacteria laced. In the presence of only antibiotic, nothing much happens. Shockwave alone, bacteria are not killed. But when you give shockwave and antimicrobial together, there is completely clean dentine surface, showing that indeed this entire proof of concept works very, very well. And now this is under manufacture, and this is under a kind of a trial by the dental colleges to understand. So it will be like a small tube which will be used to break your biofilm and save the tooth. So this is one of the most important discovery of the shockwave. The ability to treat, so we had one more challenge. So whether we can use the shock wave on chronic periodontitis, because periodontitis is again one of the most important dental disorder, and the antibiotic here also does not work. It is kind of a chronic disease, but our challenge was something different, because we could not get the work done on the human, as the ethical committee permission was extremely strict. So we had to develop a model of our own. So we developed this chronic periodontitis model in the rat. Now our MD student, he got all the samples of the chronic periodontitis pooled bacterial lysates, not lysates, but the entire saliva. And we first figured out that what all the bacteria there, the consortium of bacteria, and then we tried to inject it in this rat and try to see whether they can be made into the chronic periodontitis model. It was very challenging because it's species specific. Something you take from human and put it in the rat may clear it off, because they may have the system to defend it and clear it off. So it took really quite some time to establish this model to us, but we were very successful. You can see here that entire tissue maceration which is being caused, it is so much so that this is a very dead and lethal model, because this mouse, they die of this periodontitis. And the microbial load also is very, very high. But if you see that, if you have the shockwave and antimicrobial, look at the microbial load in this model. The rat could be completely cured of chronic periodontitis after the shockwave and antibiotic treatment. So it's a combination treatment. Successful treatment of the biofilm using the shockwave and the multi-species chronic periodontitis in the humanized rat model. These again came up as some of the really major discoveries, which will be now taken to the field very soon. Now, very interestingly, what happened, I normally will never tell about these, because we don't speak about the science. The science gets spoken by the people. I mean, you don't simply go on praising your style. But this one I never forget, because this came, our all paper work came in Science Translational Medicine, which is one of the most important journal of science, with extremely high impact factor. And they wrote editor's choice on certain articles, on our article in Science Translational Medicine, and a shocking development. It's a very interesting article. If some of you get a chance, please read it, saying that it is a very shocking development and how one can really tweak it and figure out. So of course it got a lot of publicity, etc., etc. Anyway, but now we are trying to look forward to indeed launch it so that mankind will get benefited. That is our final aim. Now, the diabetic foot ulcer. As I mentioned to you, that the diabetic foot is one of the most chronic cause of the death in the diabetic individual, and these are the non-healing ulcers. So we used a diabetic mouse model to understand that how the diabetic infection is caused and how you can use the shockwave. So this is, say, for example, is untreated mouse, and this is the shockwave treated mouse. Both are wounded here, and this is how the device looks like. So this is a mouse where the wound is, and this is the shock tunnel, shock tube. And you don't touch the mouse completely. There is a certain 1 mm gap from with the mouse skin and this tunnel, and then you pass the shock, and the shock does the job. We are trying to understand. So if you look untreated versus the shockwave treated, and these are the normal mouse, which should have the normal way of closing the wound. But if you still see the difference, at day 5 of shockwave treated versus untreated, you see that shockwave treated beautifully closed the wound, and untreated still had the wound. So we wanted to understand that what is the mechanism of this, how the shock signaling lead to the closure of the wound. Then started looking into the signaling pathway of the wound healing. So vascular endothelial cell growth factor, VEGF, angiopoietin 1, angiopoietin 2, and the TGF beta, all of them play a very important role in the wound healing. And indeed, all of them were upregulated when the mouse was treated with a shockwave. The question still remains, and that is our really very, very challenging question, that how is shock perceived by human or animal in terms of signaling mechanism? That what in the cell is taking up the shock to give rise to this VEGF or the angiopoietin or the 1 or the 2 or the TGF beta, because these are all very important molecule, but how the pressure is leading to the expression of these important molecule, that is the question. Our preliminary clinical trial done on 5 patients gave very encouraging results. So you see, this is a one picture of second day treatment, a person with a really chronic diabetic wound, and this is 70th day of the treatment. There is a serious regression of this wound area, and now the person is very hale and hearty and is walking around. So we are very happy that this person's leg got saved. These are all the small trials. Now slowly we are getting into a very large trial, and it does not end here. You can have a shockwave responsive delivery system. Say, for example, you want to encapsulate anything, in these nanoparticles, and say insulin patch. So this is a very good for an insulin patch. For just looking and a proof of concept, we use ciprofloxacin because it's very easy to see. So you have your microparticles which are loaded with the ciprofloxacin, and suppose if you give shock remotely to the system, then your ciprofloxacin should be slowly released and killing. So what you're seeing this halo zone is a killing of the bacteria, which shows that the microparticle which were loaded with ciprofloxacin release the ciprofloxacin into the system. A number of shock wave, if you see with increasing number of shock waves, the diameter increased here, showing that indeed the concept of the patch, insulin patch is, for example, you can have it on the insulin, you can have it onto a microparticle, and then you can remotely access the release of the insulin into the system by the help of this shockwave. So this work has been in fact done in the mouse model, and now we are trying to understand that how can you extend it to the human system. Then indeed we did with a diabetic mouse, and we have a model of the diabetic mouse system, used the SRES skin infection model, and understood that whether indeed the bacterial burden can be brought down. In a nutshell, the ciprofloxacin loaded particle with the shock wave only brought down the level of bacteria in the diabetic infection and not anything else. So again, showing that yes, shockwave has a potency to not only signal in the cell to somehow increase the healing molecules, but also to release the antibiotic in a very timed fashion so that even the bacteria are killed. So it's a two-way process. Your SRES, which is causing this infection, due to which your skin is not healing, will be killed, and the shock in turn will give rise to the signaling molecule like angiopoietin 1 and 2 and VEGF, which will now lead to the cell migration and the tissue will be healed. And in diabetes, it is the most critical way, because the diabetic wound are non-healing ulcerous wound. So I was talking to you about the insulin delivery. Even we measured the blood glucose level in the mouse by understanding that you can give this insulin patch in a microparticles and then try to release the insulin and measure the blood glucose level. So if you will see that, you just look at that pink level, insulin microparticle and shock wave. When you give them together, the blood glucose level never rises. It is very beautifully always lowered. Otherwise, in the diabetic mice, they rise very much over a period of time. So all these experiments were very, very, fruitful. And it also gave us a way of thought. Insulin delivery model, first the staphylococcus skin model, and also this insulin delivery model. Both these models now gave us hope that one can use the shock wave to remotely trigger and release the material as and when you want. Now, the needleless vaccine delivery, I am going to just quickly take you through this. You know that the needle stick injuries are one of the most important injuries that come up in the healthcare workers. And that is the reason that why you need to have this needleless vaccine delivery system. Which we made in the lab, and this is how the system looks like. So you have in this cavity, this is just blown up, that is why you are seeing it's not such a thick tube. There are certain dimensions to this tube, and now they look like a pen. You have the vaccine at the end of this cavity, and then this is the mouse. You simply trigger your trigger, press the trigger, and the vaccine gets impinged onto the skin. Luckily, what happens is that the vaccine get impinged to a level that it starts evoking the immune response, and it is painless because it does not go that deep. And only 160 micrometer, there are no nerve endings, so it's very comfortable. We tried using our homegrown vaccine, for which I have a patent against Salmonella typhi as well as typhimurium. We used this vaccine with this needleless vaccine delivery system, and we measured and we understood that indeed, it leads to the decrease in the challenge with Salmonella. So the needleless vaccine system works very well. We also wanted to take it up to the Mycobacterium BCG vaccination, because you know that the Mycobacterium tuberculosis is still a very major disease. And we wanted to see whether you can use this BCG vaccination from a needleless delivery system and then measure the antibody titer, and it worked very well. You see that the shockwave mediated delivery of the BCG vaccine led to really very high endpoint titer of the antibody, and it protected the mice against the challenge with the H37RA. This is the organ CFU of these system. And the post survival with the H37RV, RV is the virulent chain. So this was done in a BSL-3 lab. It's a specific lab. You can't work with MTB anywhere else. So if you see this, that all 100% of the mice survived beautifully when you gave this shockwave mediated delivery of the vaccine, that is a BCG vaccine. Yeah, so again, a lot of appreciation, etc., was there, but we are still waiting for the system to be launched. So there are now the pipeline is, our pipelines are amazing. So we have something for the lab where it's artificial transformation in the bacteria, the electroporation versus sonoporation versus heat shock. So out of all these, our system, that is by the shockwave system, not the heat, these are the classical method. All of them have their own limitations: low transformation efficiency, loss of viability. All of you must be doing some transformation experiment, and you know that how difficult and it is frustrating actually to have your DNA get inside the bacteria, and half of the time you end up with a negative result. So this, our electroporator system again gives an amazing data about the efficiency of transformation in many bacterial kinds: Mycobacterium, E. coli, Salmonella, Pseudomonas. And we worked on the mechanism, that why and how it happened. I don't think we will get into the detail of the mechanism because this needs really, I just want to give you a glimpse that there is something to do with the bacterial elongation. That is a signal once the bacteria perceive these pressure pulse, and the Young modulus law changes, and that is how in fact they take up the DNA. And this is very important. If you compare the micro shockwave device, the cell preparation time is reduced from 3 hours to 4 hours, it become growth phase independent, because in electroporator you have to only have that log phase and then hundreds of steps to make the cell competent. The cell death here is more than 75%, but here there is no cell death. The cost of the equipment, this is now it is around $7,000, and this remained the same, only $100. Cost of each experiment is 20 cents. This is $5. So if you really compare, it's an amazing system which we are very much ready to launch in the labs, for the colleges, etc. So cannot thank enough the DRDO and the Ministry of Defense and Imprint Impacting Research Innovations, because this is indeed a success story of the two labs which came together. One is a microbiology, one is a hypersonic shockwave lab, to make something which is so important for the mankind. So simply waiting now for everything to get into the pipeline and to be released in the system. So thank you very much for listening, and now I will be open to the talk.
Ma'am, there are a couple questions in the chat box.
Yes. I am going to read the questions one by one, and then we will do it. So this is a question from Susan Phillip. Ma'am, when shock waves are given in vivo, did it not affect any of the surrounding tissue? Have you studied the changes in the gene expression in mice in response to shock wave? Yes, Susan, that was actually one of the very major questions even by the reviewer, that when you give a shock wave, what happens to the nearby tissue? Because shock travels, right? The wave travels, and body is a medium. So but that is the reason we have done the histopathology of all the nearby tissues, and there is absolutely no effect on the tissue architecture of the nearby tissue, for one simple reason, that we have very carefully characterized the pressure. The Pascal, the pressure, the PSI pressure that we use, it is so low that it does the biological function, but it does not perturb the tissue. So no, there has been absolutely no change.
Darshan, the thing, Darshan, thing
Hello?
for
Hello?
a form for attendance.
Oh, sorry, Grace is talking something.
That one.
Okay, so now
feedback form. Yeah, we'll have to put it in.
Okay, so Susan, I'll continue with my answer. Now, have you studied the changes in the gene expression in mice? Yes, we have. We have done a microarray study, and it is very, very revealing experiment to see that how the shock leads to the changes in a variety of the different gene expression, which we can cluster it under the cytokine responsive elements, to the growth factors, to the certain kind of cytoskeletal elements also. So now we are trying to understand that how actually shock leads to the change, the global change in the gene expression pattern, and definitely tweak it, because that is very important for the biomedical application. So second question is by Bablin Kaur. Hi ma'am, if shockwave increases angiogenesis, then it may induce cancer, so may have more risk or harm than therapeutic ability. Bablin, see, what happens is that the angiogenesis process, you are going to do it when you are using this device for its application for healing the tissue or the wound repair, where it is a totally anoxic condition and there is absolutely no angiogenesis at all. So that leads to the stage of a gangrene. So one has to be very careful that what is it that you're using it for. Now, the pressure profile makes a big difference. It is not that at all pressure profile, the one which I use to break the dental biofilm does not cause angiogenesis. The one which is used to do transformation also does not cause angiogenesis. So the angiogenesis is only for the diabetic wound infection. Now, our work with the NIMHANS, which talks about the collaboration which we are doing on the glioma, the reduction, that is also a very, very different pressure profile, which does not affect angiogenesis. So different pressure profile have a different effect on the gene expression of certain genes. And that is the reason why this research is so tedious, because you have to really work with many different pressure profile. So yeah, if it induces the angiogenesis, now again the question is angiogenesis the sole cause of cancer? The answer is no. We know that. Now the cancer environment, must get triggered with the help of certain proto-oncogenes or certain bacteria or certain virus. You know very well that how now these human papilloma virus or Helicobacter pylori is a causative agent of cancer. So simple angiogenesis is not going to lead to the cancer, but it is a process which helps much later in making the tumor mass. So one has to be very careful when you are making this pressure profile. Yeah. So I am not seeing any more questions. Can you kindly unmute yourself and start asking questions?
Ma'am, can I ask another question?
Of course, of course. Welcome, Susan. Go ahead.
Yeah, ma'am. No,
Yeah.
I was wondering, how is it that these, do we know the mechanism by which shock waves sort of allow healing of diabetic wounds?
No, the only mechanism that we know is when we took the tissues from this diabetic individual who were treated with, who were undergoing the shockwave therapy versus the one who were not. The tissue sample suggested that there is an upregulation of the angiopoietin 1, 2, and VEGF, which are the most important factors for wound healing. So that we know that these are the reasons. Their upregulations lead to the migration of the cell and then closure of the wound. But the major question that why and how shock is perceived by the cell, which leads to the upregulation of these particular genes. That is the question that we are trying to understand now.
So is there any similarity in, the gene expression patterns, global gene expression patterns that you've found out here to other known stress signaling pathways?
See, other known signaling pathways, say for example, the moment you give some injury, there will be upregulation of these factors in the normal condition, but not in diabetic condition. Okay. Somehow in diabetic condition, there is certain repression, which does not allow allow these factors to get upregulated. And that is why the wound becomes, it remains as a non-healing ulcerous, and it becomes, in the end, it becomes a chronic ulcerous wound. Whereas in the non-diabetic, this is the common phenomena by which they are going to express the factors and heal the wound. What in non-diabetic and how this shock can do? Of course, it can do in the normal also, but it can go to that extra step to do it and trigger it in the non-diabetic individuals also. So it has, I don't think it has anything to do with the condition. It will do the same in any individual with any kind of a comorbidity. The most important thing for us to understand is that the body is a medium, and once the shock travels through the body, how in fact, so it is very difficult question to understand, because no amount of high-speed camera or any kind of system, it should be something very, very high resolution to really understand that how it is really going through and how the gene expression, maybe the gene folding, the unfolding, etc. is happening. But we are still trying to do this in an isolated gene and trying to visualize with electron microscopy to see the genome compaction. Say, okay, if you give a shock, there are certain areas of the genes which get opened up and they are ready. But even if that happens, it has to be very specific. It simply cannot go on doing random upregulation of every gene that comes in its way.
Correct. Yeah.
So yeah, these are very challenging questions and will keep us very busy.
Thank you. Thank you.
Yeah.
Ma'am, may I ask one question?
Surely, yeah.
Do you think that the shock waves generate some form of mild stress in the cells that might lead to a situation of hormesis that might lead to the initiation of the repair of the wounds?
Possible. See, anything that you give to the body or to the cells will be perceived by the cell as something which it is not used to, and you can call it as a stress. Now, the stress is something which is not taking a toll on its viability, right? Because you use that pressure profile in which the cells are very healthy and intact. So they are not killed at all. Even the bacteria, of course, bacterial cell wall is very sturdy, and animal cells what we use, they they don't have a cell wall. But the bacteria are definitely not killed at any kind of pressure profile. The human cells can get killed if you use a certain pressure profile above 5 Pascal. So when you use much below that, somewhere around 1 bar or 0.5 bar, something like that, and you try to look into the signaling pathway, the cells, they don't look distressed, but they may be perceiving it. So what you thought may be right. They may be perceiving it as a stress. Yeah.
Thank you.
Yeah.
Ma'am. Thank you so
Thank you, Debavrati. Yeah. There are no questions in the chat box. Yeah. Yeah. There are no more questions in the chat box. Anybody else would like to ask? Kindly unmute yourself. Today is the last day anyway, so it was a very that you have enjoyed all the talk. So the academies, all the 3 academies will feel very happy to know that this first online lecture series already kicked off and is going very well. No more questions? Okay, so if there are no more questions, then thank you very much. Feel free to write to me anytime that you may wish to, and discuss your scientific thoughts. Thank you so much. Thank you, Grace. Thank you, Avantika.
Thank you so much, ma'am, for the very interesting talk. The topic was very fascinating and we all learned a few new concepts. I now request Sri Lakshmi to take over.
Okay, thank you. On this story, okay, the end of the story is the new beginning for many others. On this note, let's conclude a remarkable, memorable, and knowledgeable valedictory session. I would now call upon Dr. Grace Prabhakar to give a brief report.
It's not going to. Let's go now.
Grace.
A very good afternoon to all of you. It's been a fruitful 2 days, and it is my privilege to present a very short report of what has transpired over the past 2 days. The Science Academies lecture workshop entitled Recent Trends in Microbiology and Biotechnology was held on the 12th and 13th of January 2021. The talk was hosted by the Department of Biotechnology, St. Joseph's College, Autonomous, Bangalore. The lecture series was convened by Dr. Dipshikha Chakravarti, professor at the Department of Microbiology and Cell Biology, Center for Biosystem Science and Bioengineering, Indian Institute of Science, Bangalore. There were 8 invited lectures delivered in the lecture series. Dr. Dipshikha spoke on bacterial pathogenesis, life in a vacuole, and molecular needles, bacterial injection systems. Dr. DJ Bhagiraj spoke on microorganisms for human welfare and mycorrhizal fungi for sustainable agriculture. Dr. Chandrayappa spoke on molecular dynamics of plant microbial interface and oxidative stress and overview. Dr. K.M. Harini Kumar spoke on biofuels, future renewable energy, and on molecular markers. The workshop has seen around 244 participants registered, representing institutions both in Bangalore as well as Pan-India. Some of the institutions include Andhra University, Jamia Millia University, Geetam deemed to be university, Hansraj College, University of Agricultural Sciences, Bangalore, the Neoshina University, Central University, and St. Joseph's College, amongst others. The participants have had an overall good 2 days of lectures covering a wide range of topics from diseases, human health, to agriculture and biofuels. The sessions have been streamed live on YouTube and have also been recorded and uploaded. Both the live streams and the recorded videos are available publicly on the Department of Biotechnology, GGG, SJC YouTube channel. I thank the Science Academies, St. Joseph's College, and Dr. Dipshikha for the successful conduct of the lecture series. Thank you.
Thank you, ma'am. I would now call Dr. Dipshikha Chakravarti to give close note speech for the workshop.
Thank you, Shri Lakshmi. Thank you, Grace. So first of all, let me thank all the three academies: Indian Academy of Sciences, Bangalore, Indian National Science Academy, Delhi, and National Academy of Sciences, Allahabad, for giving us this opportunity to conduct a very challenging online workshop for 2 days. My heartfelt thanks to Dr. Grace Prabhakar and her very enthusiastic initiation, which made this conference or the workshop lecture series possible, 2 days lecture workshop possible. Under these prevailing conditions, which in fact made our life indeed come in a vacuole. And as Dr. Madappa mentioned yesterday, it was something like a kind of a domesticated fatigue which has got inside us. This might be a very nice change for all of us to at least see and enjoy the lecture series. I hope that all our young colleagues and our our teachers, they have learned a lot from the speakers about the recent trend in biology and biotechnology. And they will also approach the speakers in case they need any help or they have any questions, because the dialogue should continue. It should not just stop by lecture series. Somebody should be benefited. That is most important. And it is always an upliftment. So if it just stops, then it is not of great use. So, all our very young colleagues and young students, they have to now come up. So I really hope that they will keep their curiosity on. They're going to keep that perseverance and thinking and the deep learning on, and even the determination to do science very much on, specifically the basic science on, because we should not forget that it is all about deep learning here, and it is all about the curiosity and perseverance. So I hope that through these lecture series, many of you will be benefited, immensely benefited. And we are always there to help you out. So kindly reach us, because it's not that you learn. I also learn from you. It's a two-way process. So when I interact with you, all your very fruitful ideas and thoughts will become a food for my own thought. So I learn from you a lot. So that is why we always must keep exchanging the notes and the ideas, which is an extremely good interactive platform. So again, thanks to everybody, thanks to the St. Joseph's College and all the administration in the St. Joseph's College, Dr. Grace, and the young students. Thank you so much. Over to Grace.
Thank you, ma'am. I would now call Dr. Debati Chakrabarti for the vote of thanks.
Good afternoon. Am I audible?
Yes, ma'am, you're audible.
Yeah.
Good afternoon, everyone. I am Debharati from Department of Biotechnology, St. Joseph's College, Bangalore. I would like to take this opportunity to present the vote of thanks. During this current pandemic situation, it has become important now more than ever to maintain a steady flow and exchange of knowledge. The 2-day Science Academies lecture workshop witnessed ample knowledge sharing, and this was possible solely due to the active and meaningful contributions by each one involved in this. Thus, without further ado, I would like to start by extending my appreciation and gratitude to Dr. Dipshikha Chakraborty, convener of the workshop. Thank you, ma'am. I would like to thank the eminent resource persons for sharing their knowledge with us. My heartfelt thanks to Dr. Dipshikha, Dr. Bhagyaraj, Dr. Harini Kumar, and Dr. Channa Rayappa for making this workshop successful. I would like to thank Dr. Grace Prabhakar, Professor at Department of Biotechnology, St. Joseph's College, for coordinating this workshop smoothly and successfully. I thank Dr. Madhapa, HOD, and entire Department of Biotech at St. Joseph's College for their consistent support. I thank our beloved student coordinators and volunteers, Andrea, Avantika, Oishik, Sri Lakshmi, Darshan, Isdar, Jasmine, and Aishwarya for ensuring that the workshop progressed smoothly. I thank our principal, Father Dr. Victor Lobo, our rector, Father Dr. Sweber De Silva, our registrar, Dr. Melvin Colasso, and the entire SJC management for their support. Lastly, I would like to thank each and every one of you for participating in this workshop and making it a success. Once again, thank you, everyone.
Thank you, ma'am. Thank you all. You will receive your certificate latest by Tuesday.
Thank you, everyone.
Thank you. Thank you, everyone. Thank you, Grace.
Thank you so much, ma'am. Thank you
Welcome,
for
welcome.
everything. Thank you, ma'am.
Yeah. If you need anything, please let me know. Yeah.
Sure, ma'am. Thank you so much.
Okay. Bye-bye. See you all.
Bye, ma'am. Bye.
Bye, ma'am. Thank you
Bye,
so
bye.
much.
Bye.
Bye, ma'am.
My pleasure. Bye.